Hi, welcome to the lecture on environmental adaptation of animals. Now we will continue on the hormonal control of the body and to see that, okay, what kind of interaction we have between different endocrine organs. Previously, we already learned that we have two kinds of cells that are releasing hormones. We have endocrine cells and we have neuroendocrine cells. And beside this division, we also can divide the endocrine organs that they are on the size. So they can be discrete glands, so something visible, where there are plenty of endocrine cells that are regulated at the same time. So there's a lot of hormone released at the same time. But there also can be some diffuse glands. So there can be even individual endocrine or neuroendocrine cell that is releasing some hormone. And these neuroendocrine cells, they look like neurons because they are neurons. They receive neural information from other neurons in the central nervous system. They have axon, and they use this axon to transport vesicles containing this hormone. And then in the other end of the axon, there's axon terminal, but it's a little bit modulated, so that's why it's called a neurohemal organ. And the best place to find these neuroendocrine cells is, of course, pituitary gland. The pituitary gland is quite easy to find in the brains. It's there, folds under the forebrain. And if you take a microscope picture of that, you can see clearly that it's formed from two different tissues. So there's the anterior pituitary that is closer your eyes in your head, and then there are posterior pituitary that is a little bit on the back. And in this microscope picture, you can see two completely different kind of places or organs. So on the left hand side, there's this anterior pituitary. It's red because the cells are smaller, and there's a packed endocrine cells, and you can see some holes or scarves, etc. They are the capillaries. So it looks pretty much like typical endocrine organ. And then on the right hand side, that looks like typical brain slice. You can't see practically nothing because the cell bodies are located in the hypothalamus. So only thing that you can see over here is the axons, etc. And this posterior area of the pituitary gland, so posterior pituitary gland is on this neuroendocrine. So the pituitary contains bundles and terminations of axons that are coming from the hypothalamus. So the cell bodies are in the hypothalamus, all the dendrites are there, and the synapses, also regulation of these cells. And in the hypothalamus, there are two nucleus where these cell bodies exist, and the synapses, and also the hormone synthesis happens there. Then you have quite a long distance to transport the hormone through the axon, not for the synapse, but on this ending where you can release the hormone on the circulation. And these neuroendocrine cells are releasing two peptides, antidiuretic hormone and oxytocin. And these peptides are almost completely identical. Antidiuretic hormone contains only 9 amino acids, and it starts from cysteine and ends with glycine. And as well as oxytocin is containing 9 amino acids starting from cysteine and ending on glycine, and there are only sites 3 and 8 where there's a difference. So instead of phenylalanine, there's isoleucine, and instead of arginine, there's leucine. So 2 amino acids are different. So that makes quite huge task for the antidiuretic hormone receptor or oxytocin receptor to say that, okay, which of these is which. And these peptide hormones are then easy to modulate and form new hormones. And also it's very nice that, okay, if you are tracking from different animal groups, you can see some kind of evolution. So in the arginine vasatocin, it's in non-mammalian vertebrates. There is a combination of antidiuretic hormone and oxytocin. So on the third position there's isoleucin, and on the eighth position there's arginine. So it seems to be that that is the common ancestor for these two hormones in mammals. But among the mammals there exists also some variation, like in the pig the position 8 is having lysine. So there can be some variation and causing some differences in the structure. But to compare what kind of peptides we have in non-mammals is that in the bony fish we have isotocin, and in reptiles and amphibians and birds and lungfish we have mesotocin. Again, almost completely identical. The only difference between them is on the position 4. So in the bony fish that are in aquatic environment, they have serine, and those that are breathing air, they have another amino acid in the same position. So it might be that this has been important for some reason to be on the terrestrial environment. Okay, we are releasing two hormones from the posterior part of the pituitary gland, and then of course we have the anterior pituitary. And again we have neuroendocrine cells in the hypothalamus. So the hormonal control from the hypothalamus, they are releasing hormones in the circulation, and these hormones are going only a few millimeter or less than centimeter to find the receptors in the anterior pituitary. And then they will cause that, okay, either there will be increased secretion of endocrines in the endocrine cells, or they will be inhibiting the release. And this anterior pituitary gland is quite often in medical books. It's divided in different parts: pars distalis, intermedia, tuberolaris. But the size and the position vary so much among the animal groups. So in the comparative physiology, you don't need these. Especially because in some animal groups they don't even have all these places. Everything that is released in the anterior pituitary is a peptide or peptide related hormones. So peptides are a little bit larger, so in that case it's protein, or it can be that it's having also some sugar, so it's glucoprotein. And they can have two kind of effects. Either they act directly on the target tissue, like the growth hormone, or they are controlling other tissues by other hormones, and that's why they are called trophic hormones. So we have the anterior pituitary that is releasing growth hormone, and that affects in muscle and fat cells, in increasing or decreasing the metabolic action. But it's also working indirectly via the liver. So it's in the liver cells, in the hepatocytes, they are releasing insulin-like growth factors that is increasing the growth in many tissues. Then we have prolactin. In mammals, it's easy to remember that, okay, it's related on the growth of the of the mammary glands and the milk production, etc. But it's also used in other vertebrates. So it's not only a mammalian hormone. It's related on reproduction or water and iron balance and etc. And then the third one that is having a direct effect is not in the mammals. It's a melanocyte stimulating hormone, and it does completely the same thing as the melatonin we were discussing earlier. So it affects in some animals, in reptiles, amphibians, fish, to make the skin darker. But then we have these indirect effects. And one of the things that, hormones that comes to mind in the anterior pituitary is the LH and FSH. So luteinization hormone and follicle stimulating hormone. And we all remember that, they are affecting on the gonads, and quite often they are affecting that, they are increasing the estrogen production or testosterone production, or converting androgens to estrogens, etc. And so they are usually having indirect effects in several tissues. And then we have thyrotrophin. And surprise, surprise, that's effect on thyroid gland. And then you have the thyroid gland is releasing T3 and T4, and then these Ts are having effects on the metabolism and in the growth, etc. And then last but not least, we have adrenocorticotrophin, ACTH, that is affecting on adrenal cortex. That is then adrenal cortex is releasing glucocorticoids like cortisol, and that affects on the stress responses and etc. And this adrenocorticotrophin is the same thing that we are going to discuss a little bit more detail in the next video. Thank you.